Now we start with the video 4 on the chapter 3, which is this is chapter 3, I mean the third chapter of the A2 syllabus. So this is the A2 syllabus of the biology, which is CIE 9700. In this video we're going to talk about the remaining part of the chapter, and I hope I can finish it. Number 1, we're going to talk about glucagon, then dipsticks and biosensors, and the third is the homeostasis in plants. So we start with the hormone glucagon, which is produced by the alpha cells. So I mean, if you're going to write it in words, you have to write alpha cells. Please do not write A cells. So alpha cells, and if you remember, we just talked in the previous video that there are these islets of Langerhans, and the islets of Langerhans have two types of cells, and they can be divided into alpha cells and beta cells. Now what you have to remember is that when your blood glucose falls, now when would your blood glucose fall? When your blood glucose falls is either you're exercising or you are fasting, and these days we have a lot of people who do intermittent fasting. So your blood glucose level falls, which was the normal level was between 80 to 120, and I said it maximum goes up to 200. So if your blood glucose level falls, now this would again be detected in the islet cells. In the islet cells this would be detected, and when it is detected, then we have the alpha cells. See, so the alpha cells are going to release glucagon, and the glucagon is going, if you remember, the liver cells. The liver cells had a store of glycogen, and the glycogen was the molecule which was a large insoluble molecule. So this is glycogen inside this liver cell. Now the glycogen is going to be converted to glucose, and the glucose is going to leave the cell and enter the blood. So what is going to happen is that the blood glucose, which had decreased, now the blood glucose is going to normalize, because what has happened is this store which you had, and I always say this is that large currency note like a 5,000 rupee note, and it's going to be cashed, and you're going to have glucose molecules released into the blood. So the blood glucose will normalize, because you have to remember is that glucose is your body fuel. Now of course this results in a number of it's a very complicated process by which we have to understand, and this is the diagram which I have used from your book, and we're going to talk about this in a little more detail and how this is going to result in the increased glucose. And of course the increased glucose means that the blood glucose will normalize, because it had gone down, so it will come back to these parameters by in which it stays between 80 and 120. The first thing we say is glucagon released by the alpha cells of the pancreas enters the blood and is carried to the liver cells. Now in the liver cells, what we have to understand is the cells have a phospholipid bilayer. So as all cells have a phospholipid bilayer, but amongst these phospholipid bilayer is a receptor molecule, and this receptor is this molecule here which they have shown in yellow, and to this comes and gets attached the glucagon molecule. So the shape of the glucagon molecule is going to be complementary to this receptor. So this glucagon molecule has come in the blood, and now this attaches to the receptor on the cell membrane of the liver cells. So we've got the phospholipid bilayer, and we've got this receptor protein, and if you remember, glycoproteins and glycolipids are receptors for hormones. So the glucagon molecule, so this is the glucagon molecule which is going to come and get attached. So we have the glucagon molecule here coming and getting attached to the receptor on the surface of the liver cell. Now this results in a series of reactions, and the first thing we said, glucagon binds to receptor. Then we say number 2. Now the second point is activation of G protein. So there is another structure here which is called the G protein. Why G? Because it's GTP, not ATP, is guanine. So it activates activation of G protein. Now the G protein, when it is activated, so the activation of the G protein, now that was, please remember, the first point was glucagon binds to receptor. The second point is the activation of the G protein. Now this G protein is this thing which I've drawn here, and this G protein is activated because this receptor is going to transfer this, and if you remember this in the AS level, this is called signal transduction. And this G protein is activated, then G protein goes and sort of pushes this and activates this enzyme. And this enzyme is going to cause the conversion of, we have to understand that ATP is going to be converted to cyclic AMP. So the 3 steps which we've got to remember. The 3 step, number 1 is glucagon binds to receptor, number 2 activation of G protein and enzyme, and number 3 ATP is converted to cyclic AMP. So please everybody try to memorize this. Glucagon binds to receptor. Now why glucagon binds to receptor? Because this must be water soluble, so it can't cross the phospholipid bilayer. That is why you have a receptor for it. So activation of G protein plus the enzyme. So the G protein activates the enzyme. This enzyme results in catalyzing a reaction in which the ATP is converted to cyclic AMP. Now this appears very threateningly difficult, but I'm sure it's not that difficult if you just make it sort of a point to remember it. So the first point is, I've tried to make it easy for you all. Number 1, I said glucagon attaches to receptor, then G protein and enzyme activated, ATP to cyclic AMP. Now here are some enzymes that we need to talk about, and this is called the enzyme cascade. One enzyme activating the others activating a series of enzymes and resulting in the production of something. So ATP cyclic AMP, the third point we are clear about, but now we come on to 2 different enzymes that we're going to talk about. Inactive protein kinase. So these inactive protein kinase, just like if you don't have ink in your pen and you need to make it activated, so you need to put some ink in it. So inactive protein kinase becomes active protein kinase. So we have to remember the enzyme protein kinase. Then we have to remember inactive, then this is another enzyme which is called phosphorylase kinase. So both of them are sort of PK. So phosphorylase kinase, so the protein kinase, then phosphorylase kinase is activated, and that finally activates the most important enzyme, which is glycogen phosphorylase. And the word makes sense, glycogen phosphorylase. Now the glycogen phosphorylase, when it's converted from an inactive, is going to do the final, what we want to be done is that converting the glycogen to glucose. Now of course, as I said to you, glucagon was secreted when your blood glucose levels were low. Now your blood glucose levels are low, as I said, when you're fasting. So now what is going to happen when your blood glucose level fell, glucagon released, glucagon goes and attaches, converts the glycogen to glucose, glucose enters your blood and your blood glucose normalizes. So on one side you have insulin, which is going to be produced when your blood glucose level rises. On the other hand, you have a hormone which is called glucagon, which is going to be released by the alpha cells of the islets of Langerhans. And this is going to result in your blood glucose normalizing, and you can the body can function normally. Now as we look at another hormone, which is adrenaline, which is produced by the adrenal gland. Now that also works, besides working on other organs of the body, it has receptors. So this will be the receptor for adrenaline. So we'll have a separate receptor. Now this one was the receptor for glucagon. So in the cell membrane of the liver cell, we would have receptors for glucagon, and we would have receptors for insulin, and we would have receptors for adrenaline. Now when adrenaline comes and gets attached, it's going to do the same thing. It's going to same enzyme cascade, and it is going to result in the conversion of glycogen to glucose. Now you have to remember is that adrenaline is a stress hormone, and we say it is produced in fight, fright and flight situations, in anxiety situations in which you've seen a lion and you need to run, or you've seen something very scary and sort of you're scared, and it produces that in a stressful situation. So what you need is you need more glucose in your blood, because if you need to run or if you need to do some physical activity, more glucose would be very helpful, because more glucose will be respired and more energy will be released. So the effect of adrenaline has to be also known to you, as it is mentioned in the syllabus. Adrenaline would have separate receptors, and these receptors would also result in the same story that we did earlier, is that the G protein is activated, the enzyme is activated, then there is ATP to cyclic AMP, then a protein kinase activated, then phosphorylase kinase activated, and finally the enzyme glycogen phosphorylase activated, and we have glycogen to glucose. So adrenaline is going to have the same effect as glucagon, but it will have separate receptors. Now at times as a doctor you need to ask for a urine analysis, and the patient has to give a sample of the urine, and we have to examine it in the laboratory. So if when we do some tests on it, we find out there's glucose present and there are ketones present, then of course we're suspecting that the person has diabetes. And in diabetes, as you know, insulin is not produced, and of course it results in increased blood glucose level, and that is, when it's filtered, it's not reabsorbed. So urine will contain glucose. In a normal person, even if you eat a lot of sweet stuff, it's not going to result in any glucose in the urine. So glucose and ketones, if they are present, is going to be one of the signs that the person has diabetes. Now if, God forbid, we find proteins, proteins are large plasma proteins, will never be filtered out, and even if some are filtered, which are small, they are actually reabsorbed by endocytosis in the PCT. But if we find protein in the urine, then this person is suffering from some sort of a problems with the glomerulus, and if you remember the glomerulus, there's a basement membrane. So if that gets damaged, sometimes due to bacterial infection, then the proteins will start to leak in the urine. That's not a very good thing, because you see the return of the tissue fluid is dependent on the plasma proteins which are present. So if a person is losing protein in the urine, then there are going to be problems. So number one, the person could be having something wrong with the basement membrane being damaged, or the person has some kidney infection, or in certain cases in high blood pressure, which of course results in heart disease, the person would have some protein in the urine. Now we need to discuss what are dipsticks, and dipsticks are also known as test strips, are used to test the urine for a number of different things. We can check the pH, we can check the glucose, ketones and proteins. But basically what I'm going to talk about is, if I want to check the urine of a person and want to find out if this person urine contains any glucose. Now this is a test strip, it's just like a piece of litmus paper, it's a test strip, and this test strip has enzymes immobilized on it. So enzymes are sticking to it. Immobilized enzyme, we studied that in the AS level. So the two enzymes which are attached to it are, number one, glucose oxidase, and another enzyme which is called peroxidase. So of course they would be having different active sites. So these two enzymes are sticking to this piece of paper, or this dipstick, or this test strip, and we dip it in the urine sample. We have the urine sample from a patient, and we are dipping it into that. Now when we dip it, the enzyme starts reacting. So if this person's urine contains glucose, Then the glucose is going to react with these two enzymes, this enzyme and this enzyme. And it is going to result in the formation of gluconolactone and hydrogen peroxide. Now, also on the test strip, we have a chemical on it. The chemical is going to react, and it's a colorless chemical, it's going to react with the hydrogen peroxide. So this H2O2, which is the hydrogen peroxide, is going to react with this colorless chemical and is going to result in a brown color. Now this brown color, of course, has got different shades. The more the glucose that is present, the darker the color. So we have a color chart, and we're going to match it with a color chart, and then we're going to compare it, and we're going to say, okay, this person either has no glucose or he has 1%, 2% or 3 or different values. We have different units for it. So we're going to compare it with this color chart, which is going to give you different shades of brown, and you will then know if the person's urine contains any glucose or not. So this is what we use dipsticks for, to check if a urine sample contains any glucose or not. Now what we have to understand is what is a biosensor. Now biosensor is usually used to detect the blood glucose levels, and people who are diabetics usually have these machines at home, and they're very simple to operate. And of course this is the field of biomedical engineering, which results in manufacturing these machines, and that is what is the next careers that you should be thinking about when you're studying biology. Now the biosensors also is a strip, a test strip, which is going to have some enzymes fixed onto it. So this is again an example of immobilized enzymes, and this time you have the enzyme glucose oxidase, which is present on the test strip. But what you need to add to it is a blood sample. So you prick your finger and you need to add just a drop of blood to this. So when the drop of blood is added to it, and then you put it in a machine, and this machine is going to give you a digital readout. Now what happens actually is, so the enzyme is present on this test strip, and to this there is this little area which has actually got a pad, and on this is the enzyme, and then you add the blood to this little portion of the test strip, and then you put this test strip in the machine. So you put this in the machine, and blood contains glucose. Now that glucose is going to react with the glucose oxidase, and it is going to result in the formation of gluconolactone. Now if there is say 100 glucose, so 100 gluconolactone produced. If there's 200 of that produced. I'm just giving you an example. Now this is going to generate a tiny electric current. So a tiny electric current is generated. Now this current is detected. Now this current is detected by an electrode. Next point is it is amplified, and this is then given to you as a digital readout on the machine. And the people who are diabetics will check their blood in the morning and say, well, this was my glucose level, 120 or 130, and then they'll check it in the afternoon and they'll give you some reading, and then they'll check it in the evening and they'll give you a reading. And this of course helps the doctors to help them regulate their medication and see how much medicine do they need, are they going fine, or they need more of the medicine or not. So biosensors are little strips on which the enzyme glucose oxidase has been stuck onto it, and then you add a blood sample, and this reacts and you get gluconolactone, and a tiny electrical current is generated, is amplified, and you get a digital readout. So this is what we have about biosensors. Now the last topic to be discussed in this chapter is homeostasis in plants. Now when we look at homeostasis in plants, we have to understand that it is to maintain constant internal environment. It's just as important for plants as it is for animals. So the mesophyll cells require a constant supply of carbon dioxide if they are going to use the light energy for photosynthesis, and we can see how less carbon dioxide is going to limit the rate of photosynthesis. So stomata control the entry of carbon dioxide into the leaves, but also they are important because water vapors are going to be lost through the stomata. So in order to allow CO2 to enter and stomata open, so that is why we say transpiration is the inevitable consequence of gas exchange. Now stomata actually is made up of, there are two guard cells, and the guard cells are going to sort of increase in volume, and then they're going to, the inner walls are going to move apart, and that is going to result in the stomata opening. So we've got to understand that how does this happen. Now what we find to our utter surprise is that stomata show a daily rhythm. I mean, whether they are in the dark or light, they sort of rhythmically open and close, and you have this graph which shows you that they, even though we keep them in the dark, even then they open and close rhythmically, might be a little lesser. So they open and close even when kept in constant light or constant dark, and these rhythms persist. Of course, opening during the day maintains the inward diffusion of carbon dioxide and the outward diffusion of oxygen. But it also, of course, results in water vapors being lost. Now as you look at how the stomata respond to changes in environmental conditions, now stomata open in response to two things. Number one, increasing light intensity. Now in the morning, as soon as the sun comes out, the light intensity keeps on increasing, so stomata will start to open. Number two, they will open when there is less carbon dioxide here. Now you know this air in the air spaces. Now I said less carbon dioxide in the air spaces in the leaf. Now the air spaces in the leaf, you know, has air. But when there is a little light, what is going to happen is that the chloroplast present here, the chloroplast present in the palisade mesophyll and some in the spongy mesophyll will start to photosynthesize. So when they start to photosynthesize, they'll take up the carbon dioxide in these air spaces. So the carbon dioxide, when we said there is a low CO2 concentration. So as by morning time, when at dawn sun comes out and there's a little bit of light, little photosynthesis means the carbon dioxide is going to be used up by the chloroplasts, and photosynthesis is going to start. So maybe the photosynthesis is not that fast, the rate is very slow, but even then that is going to result in this air which had carbon dioxide decreasing. So the carbon dioxide concentrations will decrease. So low carbon dioxide concentrations is going to cause the stomata to open, and that is very important. Why? Because then the carbon dioxide is going to diffuse in, because there has to be a gradient, there has to be more carbon dioxide outside and low carbon dioxide inside. So that is why there is going to be diffusion of carbon dioxide, and of course that is going to result in the production of oxygen, and the oxygen is going to diffuse out. So what we have to understand is that carbon dioxide entering the leaf is important for photosynthesis, because that is a limiting factor in photosynthesis. But then as the stomata open, the plant is going to lose water in the form of water vapors by transpiration. So that needs to be avoided, but we cannot avoid that. That's an inevitable consequence of gas exchange. Now stomata close in response to, number one, we've said darkness. Second, naturally during darkness, when the plant is only respiring, so there's no photosynthesis, that is going to result in a high carbon dioxide concentration inside the air spaces of the leaf. So that is also going to cause the stomata to close, which is again a safe mechanism, because now photosynthesis is not taking place. So darkness, number one. Second, high carbon dioxide concentration, low humidity. Now you've got to understand that in a leaf, if there is less humidity outside and there's more water vapors inside, now these water vapors are going to diffuse out. As you know, water vapors are always more inside the leaf. And if there's less humidity in the environment, in the air outside the leaf, so there's going to be a very sharp gradient, and all the water vapors are going to diffuse out. Now this of course is going to result in a lot of water loss, and the plant could wilt. So if there is low humidity outside the leaf, less water vapors outside the leaf, less water vapors, please remember I'm not saying water, I'm saying water vapors outside the leaf. So less humidity outside, even then the stomata is going to close. So number 1, darkness. Number 2, high carbon dioxide concentration. Number 3, low humidity. And the fourth one is high temperature. Now if there's going to be a lot of very high temperature outside the plant, what is going to happen? This is going to result in more evaporation, more evaporation, more transpiration, because the rate of diffusion will increase. So more evaporation, more transpiration. This of course again could be disastrous for the plant because it would lose too much water. Just like if you sweat a lot, you'd lose a lot of water and salts. So if there is high temperature, even then the stomata are going to close. And the fifth and the most important point is water stress. You see, if there is no soil water, then the plant only gets water from the soil through the roots. But that water is in the soil, not in the roots. So the soil water, it is called water stress. If there hasn't been rain for a long time, the soil is really dry. Now the plant can wilt. So if there is water stress, stomata will close. So if there's no rain maybe for the last 10 days or 15 days and then suddenly there is water, the plant will recover. So stomata close in response to number 1, darkness. Number 2, high carbon dioxide concentration. Naturally, if inside the leaf there's no photosynthesis, so CO2 is going to increase because the plant will respire all the time. Only during the day what happens is photosynthesis is more than respiration. So high carbon dioxide concentration inside the air spaces of the leaf. So this high CO2 concentration is, we're talking of the air spaces in the leaf. Air spaces in the leaf. Right? Then I said low humidity. Fourth was high temperature, which I've just explained, more evaporation, more transpiration, and water stress when there is no water in the soil, so the roots are not, no water is moving into the root hair cell and no water is moving up the xylem, and so the plant is stressed and it actually releases a hormone called abscisic acid. And this abscisic acid is what we're going to study in the next video, and this is also called ABA, abscisic acid, which results in stomatal closure. So this ends this video, and there's going to be another video which is going to address how the opening and closing of stomata takes place. There's a very short video after that, and that will finish this chapter. Thank you everybody.